So good morning, everyone. It's a great honor for me to start this meeting. And before starting, let me thank the organizer and Federico for the invitation. So as you heard, today I'm going to talk about the transcription factor MITF, simulation, senescence in melanocytes, but also in melanoma cells. So first, a few words on senescence. So senescence is referred to aging and age-related disease such as cancer. This is a major tumor suppressor mechanisms that stop many cancers just as they start to grow. And this process is so important that Elizabeth Blackburn got the Nobel Prize in 2009 for her work on senescence. So senescence is said when cells exit the cell cycle and stop proliferating. And this proliferation arrest is not responsive to mitogenic stimuli, which is different from quiescence. So senescence can be replicative or premature. Replicative senescence is associated with telomere shortening every time a cell divides. And this is what is called the Hayflick limit, reported by Hayflick and Morehead in 1961. Senescence can also be induced prematurely in response to stress. This is stress-induced senescence, or oncogenes. This is oncogene-induced senescence. And this senescence, this premature senescence, is telomere-independent. So both types of senescence are morphologically indistinguishable and display similar markers, such as cell flattening and enlargements. They become positive for the senescence-associated beta-galactosidase activity. They display senescence-associated heterochromatin formation that can be detected by, for example, the heterochromatin protein 1. They display activation of the DNA damage response inhibiting pathway that can be detected by 53BP1 or activated form by phosphorylation of ATM and CHEK2. They can also display activation of tumor suppressor pathways such as P16 and P53. And finally, they can also produce an inflammatory secretum. So from melanocyte to melanoma, the process of senescence seems to be central in the melanocyte lineage. So in melanocyte, so you know that several pigmentary changes occurs during aging or photoaging because UV is a well-known skin aging factors. So here, this is a picture of senile lentigo, which is also called solar lentigo or H-spots. That is hyperpigmented spots that often develops on sun-damaged skins of the face or on the back of the hand, for example. The nevus is one of the most prominent examples of senescence in vivo. It is thought to be a form of melanocyte that enters the process of senescence following oncogenic activation, such as oncogenic BRAF V600E and NRAS Q61K or HER, which are the two main mutations identified in melanoma. And here, this is senescence-associated beta-galactosidase staining of a nevus, and you can appreciate this typical blue nevi in this lesion, indicating that the nevus are in a senescent state. And finally, several mutations, whether at the somatic or germline, in melanoma are related to senescence or the control of cell proliferation, which is CDKN2A, which encodes for the tumor suppressor P16, the retinoblastoma protein, or the telomere maintenance genes, SPOT1, HCD, TER2IP, and TERT. Another factor that is important in the melanocyte lineage is MITF, which stands for microphthalmia-associated transcription factor, and this is the master gene of melanocyte of homeostasis. So MITF is a basic loop helix leucine zipper factor, which binds as a dimers on specific sequences called E-box localized in the promoter of its target, in the locus of its target genes. And here, this is a mouse with MITF invalidation. And you can see that this mouse is white, and it is white not because it has non-functional melanocyte, like in albino, but because it has no melanocyte at all. And there is also some MITF mutation that can be detected in patients suffering from the Waardenburg syndrome type 2A. So MITF is also important for the differentiation and pigmentation process. And years ago, when I was PhD student, we showed that the cyclic AMP elevating agents, so here for scoline, this is cell pellet and in vials. And so you can see that cyclic AMP elevating agent stimulates melanin synthesis. And when we use a dominant negative form of MITF, the melanin synthesis is dramatically decreased in response to cyclic AMP elevating agents. And finally, MITF is also important for melanoma. And again, we show here, we identify the first pro-oncogenic mutation in MITF that predisposes carrier to melanoma, and we will talk about this mutation later on. So we don't know precisely how MITF controls these different biological functions, but a model proposed that the activity of MITF depends on its expression level, on its post-translational modification, among which simulation, and its cofactor bindings. And this creates a sort of barcode-like situation that channels MITF towards a subset of specific target genes and drives a different biological function. So today we will talk about simulation and senescence, as I previously told you. So because I show you that MITF is important for the development of the melanocytic lineage, and because MITF is also important for the proliferation of melanoma cells. Years ago, we thought that MITF was a good candidate to pursue as a target to control the proliferation of the melanocytic cells. And so for that, we used the approach of RNA interference to inhibit MITF expression. And this is what you can see on this Western blot. This is a MITF-specific siRNA that efficiently inhibits MITF expression. So of course, we worked with several siRNA to rule out non-specific effects, but today I show you data with only one siRNA. And so, for simplicity. And so what you can appreciate on these pictures is that in response to MITF knockdown, cells become flattened and enlarged. They display this typical blue staining that reflects the senescence activity. They also show these dots of heterochromatin protein 1 gamma, and this is the formation of senescence-associated heterochromatin foci. And here, you can see also that compared to control cells that proliferate over time till 6 days, here in these experiments, here the curve is flat, so MITF knockdown cells stop proliferating, and they are actually arrested in a G0, G1 group phase. We also assess the induction of apoptosis or cell death. So on this Western blot, you can appreciate that in contrast to TRAIL, which is a pro-apoptotic ligand of the TNF-alpha family that we used as a positive control here in these experiments. So TRAIL induced caspase activation, caspase or cysteine protease, which are activated during the process of apoptosis. So this is here the doublet. That is the active form of caspase-free, and here this is a cleavage of PARP, the polyADP ribose polymerase, which is one of the best known target of caspase-free. And so you can see that there is caspase activation and cleavage of PARP in response to TRAIL. However, we cannot detect caspase activation or PARP cleavage in response to MITF suppression knockdown after 3 or 4 days. We also performed some FACS analysis, and we didn't detect any sub-G1 peaks, which reflect cell death after 3 or 4 days of MITF knockdown. So MITF knockdown cells display hallmark of cellular senescence, but no sign of apoptosis of cell death. So I also told you in the introduction that senescence is often associated with activation of the DNA damage response-signaling pathway. So this is something we looked to. And so for those who are not introduced, very briefly, DNA damages induce the recruitment of sensor proteins such as the activated form of H2AX called gamma H2AX and 53BP1. that creates a sort of platforms to recruit effector proteins, such as ATM and CHEK2, that with P53 coordinate the cell cycle arrest and DNA repair, if it is possible, otherwise cells enter senescence or apoptosis. And so on these immunofluorescence experiments, you can see that a MITF knockdown triggers DNA damages, and the recruitment at site of damages of gamma H2AX, 53BP1, and the activated form of ATM and CHEK2 that are phosphorylated here and recruited at this site. What you can appreciate also on this Western blot is that a MITF knockdown is associated with the phosphorylation of P53 and serine 15, which induce P53 stabilization, and indeed we found an increase in P53 level in cells with a MITF knockdown. When we used inhibitor of the DNA damage response signaling pathway or a siRNA against P53, we dramatically inhibit the ability of melanocytic cells to become positive for the senescence-associated beta-galactosidase mediated by a MITF knockdown. So the DNA damage response signaling pathway is involved in the senescence program mediated by a MITF knockdown. What is interesting also to note is that this is something that we observe in our cell culture. So this is confocal images with the 3D reconstructions. So you can see that in control cells, chromosomes that are detected with phosphohistone H3 here in red, they are properly aligned along the equatorial plates. At the telophase, you can see here the two pool of sister chromatids that seems to be evenly divided between the two daughter cells. And here at the cytokinesis steps, you can see the mid-body that seems to be properly formed, which is detected with beta-tubulin in green. And in a MITF knockdown cells, you can appreciate here some chromosomes that lags, that are not correctly aligned along the equatorial plates. You can see that we detected at the telophase here some chromosome lagging between the two daughter cells. And when cells finish to separate, this chromosome lagging are sometimes excluded into micronuclei. So we counted the micronuclei, and we indeed found an increased number of micronuclei in the cells with the knockdown of MITF compared to control cells. And this is a sign of genomic instability. And finally here at the cytokinesis step, you can see that the beta-tubulin is not localized at the most apical site of the mid-body, and so this will lead to defect aberration in cytokinesis. So to determine how MITF controls these processes, we combine an approach of RNA-seq experiments of MITF knockdown versus control cells with a genome-wide chip-seq of MITF. So this is an example of those two experiments picture. And this, the combination of those two analyses allowed us to identify the full repertoire of all MITF direct and indirect target genes. And among the MITF direct target genes that are positively regulated by MITF, we found genes associated with cellular functions such as cell division, DNA repair, and DNA replication. So to sum up this part, this is a scheme that I would like to propose. So MITF knockdown trigger mitosis defects, the formation of nucleoplasmic bridges and micronuclei that leads to DNA damages and activation of the DNA damage response. I told you also that MITF positively controls some genes involved in DNA repair and replication, and so MITF knockdown should dampen this, the efficacy of the DNA damage response signaling pathway, probably amplifying some DNA damages that can also trigger some mitosis defects, and these damages converge towards P53 induction and senescence entry of melanoma cells and melanocytic cells. So this brings me to the second part of my talk on the MITF mutation that we identified. And to make a long story short, how we go to identify this mutation. So years ago, we identified MITF as a new substrate of caspases during apoptosis in melanocyte and melanoma cells. So what we did is site-directed mutagenesis to identify the caspase cleavage site in MITF. So we identify the caspase cleavage site. We so generated a non-cleavable form of MITF. We forced the expression of MITF in melanocytic cells, melanoma cells, actually. And what we observed is that the non-cleavable form of MITF render melanoma cells resistant to apoptotic stimuli. And so this prompted us to hypothesize that individuals who develop melanoma harbor a mutation in the caspase cleavage site, in MITF in melanocyte, leading to melanoma formation. And so we collaborate with a team in Paris to sequence MITF. So far we haven't identified a mutation at the caspase cleavage site, but we identify a mutation near this caspase cleavage site that changes the glutamate creating into lysine. So this is our study. So we found that the frequency of this variant is higher in patients who develop melanoma, whether single or multiple melanoma, compared to controls. So this variant has also been identified by other studies that are listed here with similar allele frequency. They also found that the variant, the MITF mutation, is associated with melanoma, whether single, multiple, but also familial cases of melanoma in this study, and with nodular melanoma in this study, which is a form of aggressive melanoma. So this variant confers a 2 to 5-fold risk to carrier of developing melanoma. And what is interesting also to note on this slide is that the MITF variant has been associated with a higher nevus count and fair skin in 2 studies, which are risk markers for melanoma. So of course, the first things we did is to determine the impact of this mutation on MITF. And so again, this is a schematic representation of MITF with all the post-translational modification reported. And 2 simulation sites, K182 and K316, were previously reported. So the glutamate creating is highly conserved among species from human to fish, and falls within the second simulation site here. And so the mutation changed this consensus simulation site, IKQE, into IKQK. That means that probably the mutation impact on the simulation of MITF. And this is indeed what we demonstrated. So here, this is a representative experiment performed in HEC293 cells in which we force the expression of MITF because MITF is not expressed in HEC293 cells. And we also force the expression of SUMO. So here, this is MITF that is detected as a doublet. This is a lower exposed blot of MITF. So when we force the expression of SUMO, we detected the simulation of MITF here and here. When we introduce a mutation of the first simulation site, we decrease the simulation of MITF. When we introduce the mutation, we identify in patient, we even more decrease the simulation of MITF. And we perform the double mutant, we abrogate the simulation of MITF. So more recently, we confirmed this data, this observation, in a more physiological settings, without forced expression of SUMO or MITF. So we perform proximity ligation assay in melanocytes that we isolated from patients with a wild-type form of MITF or from melanocytes with a mutant form of MITF. And so here, the red dots represents the association of MITF and SUMO. And this is what we detected in melanocytes with a wild-type form of MITF, but not in those with the MITF mutation. So having shown that the E318 lysine mutation impairs the simulation of MITF, the next question we had addressed is to determine the impact of this mutation on the cellular localization, stability, activity of MITF. So we found no change in the cellular localization or stability of this mutant form of MITF compared to the wild-type form, but what we observed is a change in its transcriptional activity. So this is a very simple experiment shown here that use a synthetic reporter vector that comprise 3 E-boxes, which are the MITF binding site, cloned upstream the luciferase genes. So what you can observe here is that when we co-express the wild-type form of MITF or the forms with a mutation in the first simulation site, we have similar transactivation activity of MITF, but when we introduce the mutation we identify in patient, we strongly stimulate the transcriptional activity of MITF. And this is in agreement with the well-known dogma of sumo-mediated transcriptional repression. So because this mutation stimulates the transcriptional activity of MITF, and because MITF binds to DNA, we perform a genome-wide analysis of the wild-type form of MITF and its mutant form. And so what this experiment shown here told us is that the wild-type form of MITF bind to about 9,000 loci, and the mutant form to about 22,000 loci, and the difference being a large number of weakly occupied sites by the mutant form of MITF. So this comparative data analysis also show that some sites are equally occupied by the wild-type or mutant form of MITF, such as in cluster E, for example, and some sites are preferentially occupied by the mutant form of MITF, such as in cluster D or C, for example. On the right here, you have some example of the preferential occupancy of the IF1 alpha, AP1, and ABCB5 loci by the mutant form of MITF compared to the wild type. So I show you that E3 18 lysine mutation triggers MITF hyposimulation, change its genomic distribution, and change its transcriptional activity. So to assess the how MITF is related to the pathogenesis, how this mutant MITF is related to the pathogenesis mechanisms, we constructed a new Knokin mouse model harboring the MITF mutation from its own promoter. So these mice do not show growth abnormalities, no nevi or melanoma development within 24 months of age. The only phenotype we observed is a slight lightening of the color of the coat, the hair, and the tail. So this is here an example of Fontana-Masson silver staining of skin tail. So here in the mice with the wild-type MITF, you can appreciate the black melanin here detected by the staining. That are strongly reduced in the skin of MITF with the MITF mutation, and this is on the right, the quantitation of melanin. So this finding is actually in agreement with what was reported in some studies. I mean the association of the MITF mutation with fair skin. So next, what we did, we started to introduce the MITF mutation in 2 mouse models prone to develop melanoma. So the first model we used is a model that allows expression of oncogenic BRAF, specifically in melanocytes, thanks to the tyrosinase promoter after tamoxifen application. So this oncogenic BRAF mouse is nevogenic, it's known, and this is indeed what we observe. These mice develop nevi. But when we introduce a mutation of MITF, we significantly increase the number of nevi in this BRAF background, indicating that MITF is nevogenic. And again, this is in agreement with what was reported in some studies. I mean the association in human of the MITF mutation with a higher nevus count. Regarding melanoma development, you can see that these mice develop melanoma with a very long latency. It's about the blue line here, 400 days for the first mouse. And we found no change in the latency nor in the penetrance when we introduce a MITF mutation in this background. So what we did next is to introduce a MITF mutation in the second mouse model prone to develop melanoma, which allows the expression of oncogenic BRAF and loss of PTEN specifically in melanocytes after tamoxifen application. And here, this is a representative kinetics experiments showing that melanoma develop faster in presence of the MITF mutation. And for example, you can see here that after 26 days, in the mice with the wild-type form of MITF, they still show nevi that are very well separated, very well distinguishable. But here, those nevi already grew a lot and gather and form these tumors. So above, this is a Kaplan-Meier survival analysis that confirms this observation, showing that mice with a MITF mutation display shortened survival times compared to mice with the wild-type MRTF. So to determine at the molecular level how the mutant form of MRTF mediates its effect, we perform a transcriptomic analysis of 4 different tumors from those 2 groups. And what Ingenuity pathway analysis told us is that the MRTF mutation is associated with a decrease in expression of genes that are related to senescence, such as CDKN2A, CDKN2B, and CDKN1A. And this indicated that the MRTF mutation decrease the process, the ability of cells to enter senescence. So here, this is the hypothesis we tested. We used human normal melanocyte isolated from patients with a wild-type form of MRTF and with a mutant form of MRTF, in which we force the expression of oncogenic form of BRAF. So you have here the expression of the oncogenic form of BRAF, which translated into a similar activation of ERK downstream. And what you can appreciate here is that in a melanocyte with wild-type MRTF, the oncogenic form of BRAF is able to induce this typical blue staining that reflects senescence entry of melanocytic cells. But melanocyte with a mutant form of MRTF display a delay in their ability to enter the senescence process. So the MRTF mutation seems to impair oncogene-induced senescence in normal human melanocyte. So to sum up, this is the schema I would like to propose. So melanocyte can give rise to nevus or senile lentigo, and this is related to senescence, premature senescence, replicative senescence, or aging. And our work showing that MRTF inhibition can trigger senescence could be of interest in this process. And more globally, we identify the full repertoire of MRTF target genes, among which we find some genes that regulate pigmentation and the senescence process. And our finding can also find interest in the progression of those lesions to melanoma. So I told you that nevi are considered the very best example of senescence in vivo. They can remain static for decades and never progress towards malignancy unless other alteration occurs. That leads to senescence bypass. And with this senescence bypass, cells restart their proliferation with less checkpoint control, so genomic instabilities occurs. And with reacquisition of pseudo-epithelial mesenchymal transition, cells start to disseminate, form mets, and usually these metastases are drug resistant. And so our finding could find interest in these 2 processes to improve anti-melanoma therapies. So to finish, I would like to thank our sponsor, and I would like also to thank all the people in the team who, and some who participate to that work. And I would like, also, of course, to thank Robert. And this is here our collaborator in France or abroad. Thank you very much for your attention.
Thank you very much, Corinne. Is there any question?
The first mice with the mutation, you said that this mice has some code color dilution. And if I understood well, these mutants have some more transcriptional activity, more binding to targets. So I'm not sure to understand the relation between the 2. So if you can just comment on that.
Yeah, okay. So regarding the first question, we have lots of genes involved in kinetochore in the list we identified by our RNA-seq and chip-seq analysis of MRTF. Okay. So for that, it's we don't have only one. So we have several genes involved on those process of replication and the DNA repair and kinetochore formation, etc. So yes, this fits with the phenotype we observed. Regarding the regulation of pigmentation by the MRTF mutation, it's true that we observe an increase in transcriptional activity, but it's a transcriptional activity, it's a global transcriptional activity on on e-boxes that are not in their environment, genomic environment. And so we know that MITF is able to bind to genes that are not related to pigmentation, but that can affect pigmentation. And we know that MITF can bind to DNA and exert some repressional transcriptional activity. So I guess there is a mix that we don't know for the moment between those activities that leads to a decrease in pigmentation. But yes, actually, Cedric, the regulation of pigmentation is not huge. It's a slight lightening. So we don't know because we didn't succeed at the lab to stain, to know if it's a change in the activity of melanocyte or in the number of melanocyte in the skin. So I have no answer for the moment to that.
Corinne? Perhaps there are something I do not understand exactly, but you have shown that the mutant MITF delay senescence induced by RAS or RAF.
BRAF, yes.
In vitro, but in vivo, you have shown that there are more NEVI. Is it correct?
Yes.
So I don't really understand. In vitro, it delays the occurrence of senescence, and in vivo, it induces more NEVI.
Yeah. Because it's true, you're completely right. We are I show two things. I show that indeed there is an increase in the number of nevi, and what we show is that the MITF mutation, the melanocyte with the MITF mutation, have a doubling capacity that is higher than normal melanocyte. Okay, we have that. So they proliferate a little bit faster, and I think that's why we saw an increase. But after that, they are able to stop. There are other checkpoint control that allow them to stop. So indeed, in the oncogenic BRAF background, we show that. In the BRAF PTEN background, we show that nevi actually they progress towards malignancy, towards formation of melanoma, and they don't stop because you lose also another checkpoint here, and so they are able to move towards melanoma. And in the culture conditions that we used, we also have this increased activity in the PI3 kinase pathway in the medium that we used with the MITF mutant, and we think that that's why we observe this ability to delay the capacity to enter senescence.
Very nice presentation, many thanks. I was wondering about the influence of the simulation versus a potential ubiquitination at that site, and if that would also impact on the stability of the protein on the one hand, and that then would lead to a second question about have you ever tried to overexpress MITF in melanocytes, and would that then be prolonging the replicative lifespan and be protective? Could that be a target for increased activity as an anti-tumor mechanism?
Okay, so for the first question, you're right. So the mutation changed glutamate into lysine, so it's not nothing, and we lose the simulation for sure, but perhaps it's not directly the simulation impact that affects all the processes we looked at, but perhaps it's a change to ubiquitination. You're completely right, and we haven't studied these mechanisms, so we don't know, but we know that simulation is affected by the mutation. And for the second question, yes, we also have a delay in the senescence when we force the expression of MITF, but you can have also regulation of simulation independently of the mutation. So. But the senescence bypass is higher with the mutant form of MITF.
Okay, I have just a short question for you before you leave. I'm wondering if you, in the list of your protein interacting with MITF, you don't have lamine A or lamine C, because in diseases with mutation of the gene LMNA encoding these proteins, you can have a trouble of pigmentation. You have hypo and hyperpigmentation. So, and you have also alteration of the nuclei of the cells. So I was wondering if you have found this protein.
I don't know I don't know precisely the list of all MITF target genes, but I can check, and there are many nuclear proteins. So perhaps they are lamine C and A.
And A, yes.
I can check for you if you want.
Okay, thank you very much.